I'm Sakib Huq, and I've been working in Kash's lab in the Bowles Center for Alcohol Studies. And the name of my project is the role of the Dynorphin Kappa-opioid receptor system in mediating the stress response in mice. I'll start today with some of the relevance of this work from a public health and academic standpoint, and then talk about two main technological approaches I used and several behavioral assays to manipulate and test these mice. So the ultimate goal of this research is to better understand some of the mechanisms involved in the stress response and anxiety disorders. And this is really relevant from a public health perspective in that anxiety disorders are the most common mental health illness in the United States, affecting over 40 million adults a year. We know that this is very costly to our healthcare system, and we also see a high comorbidity between these anxiety disorders and other psychiatric disorders and alcohol and drug use disorders. We also know that stress is a potential cause or contributor to these anxiety disorders, as well as several other issues throughout the body and the mind. Now, there's been several brain regions that have been implicated in these disorders, and one that we're interested in the Kash Lab is the beta nucleus of the stria terminalis, or the BNST. The BNST is a band of fibers running along the lateral margin of the ventricular surface of the thalamus, and it's a critical relay station in the brain. It's been shown to be a major output pathway of the amygdala, and it also has projections going to and from several other brain regions that you can see here, and these regions are known to be involved in anxiety as well as drug-seeking behavior and alcohol disorders. Now, in the BNST, we see members of the endogenous opioid system expressed, and this system is known to mediate emotional and behavioral responses to stress. And among those broadly expressed is the Kappa-opioid receptor, which I'll call CORS, and its endogenous ligand Dynorphin. Now, CORS are known to exist in several regions throughout the brain, again, several of which you can see here. And previous work has shown that binding of Dynorphin to CORS has been able to induce an anxious, stressful phenotype in mice. Now, most of the work that's been done has looked at the effects of CORS systemically, but we're especially interested in CORS in the BNST. Now, here's an image that I took from a Dynorphin reporter mouse, where in green you see a fairly high density of Dynorphin cell bodies here in the BNST. Now, again, we know that Dynorphin is the endogenous ligand for CORS, and we already knew that CORS exists in the BNST. So basically, we know that this is a relevant and interesting region to be looking at for this work. So the general idea of what I did was to use two technological approaches to manipulate Dynorphin neurons in the BNST, and then run mice through several behavioral assays to see if we could produce a different phenotype. The first of those two was to selectively ablate the Dynorphin neurons from the BNST, and the second was to functionally inhibit them by altering their excitability. Now, we hypothesized that ablating Dynorphin neurons or functionally inhibiting them in the BNST would lead to a less stressed, less anxious phenotype in these behavioral assays, which I'll discuss shortly. So the first of those two methods involved me stereotaxically injecting an inducible Cas-based construct into the BNST of Dynorphin-carene mice. So basically, using this equipment here, a stereotax and computer software, I was able to very precisely inject this Cas-based construct into the BNST. And the idea is that this construct selectively induces cell apoptosis in Cre-expressing neurons. And since we're using Dynorphin-carene mice, this is essentially causing apoptosis of Dynorphin-expressing neurons in the BNST. Now, I also did a group of control surgeries where I injected channelrhodopsin into the BNST. This was just a control for the surgery itself. The channelrhodopsin doesn't actually have any effect. I then ran the mice through three different behavioral paradigms, and I ran them in the order shown here. First was the elevated plus maze, or EPM. Next was the open field, and finally was the forced swim stress assay. So first, with the elevated plus maze, as you can see it here, this is basically a maze about 1 meter off the ground with 4 arms. 2 of the arms are closed, meaning that there are walls enclosing the arm, so a mouse on that arm can't possibly fall off. And 2 of the arms are open. Now, this is kind of the equivalent of walking the plank in that they aren't enclosed by any walls, and that the mouse could fall off at any given time. So the mice are able to freely roam for 5 minutes, and we look to see how much time the mice spend in the open arms versus the closed arms, and how many entries they have into the open arms, because this is indicative of a less stressed, less anxious phenotype. Now, in this first figure here, we're looking at the time spent in the open arms of the elevated plus maze. Sample sizes are shown on the bars, and we're looking at our control and Cas-based mice. And as you can see, we don't see a difference between the two groups. So I then moved the mice to the second assay, which is the open field. This is just a box that's about 50 centimeters by 50 centimeters, where the mice are able to freely roam for 30 minutes. And then we look to see how much time the mice spend in the center of the chamber versus the corner of the chamber. Now the center is more exposed and out in the open, and so it's known that mice that spend more time in the center are showing, again, a less stressed, less anxious phenotype. So again, in this figure here, we can see we're looking at time in the center between our two groups, and again, we don't see a difference between the two groups. So next I moved the mice to the forced swim stress test. Here the mice are placed in a plexiglass cylinder filled with water and forced to swim for 5 minutes. And we look for one of two states. They're either mobile or immobile. And when they're mobile, the mice are actively swimming around attempting to stay afloat. And when they're immobile, they've basically stopped their movement and are just floating on the top of the water. Now this has actually been used as a really great screen for depressive phenotypes, with the mice that spend more time immobile showing this more depressive phenotype. And as you can see from the figure here, we're looking at the percent time spent in immobile. And again, we don't see a difference between our two groups of mice. Now, it's important to note that the first two assays I showed you, the elevated plus maze and the open field, were done under basal non-stress conditions, and that the tests themselves really aren't particularly stressful. So next we wanted to see if we were able to produce a difference in phenotype in the mice using these assays under stress conditions. So what we did here was a new paradigm where we used wild-type mice. These were not Dynorphin-crees, and we didn't perform any surgeries on them. But we used wild-type mice and put them through the forced swim test, and then immediately placed them on the elevated plus maze after a very brief recovery period. The idea was that the forced swim could serve as a significant stressor, and we wanted to see if we could see a phenotype. So when we look at the time spent in the open arms for the forced swim EPM group, there are two groups. One is the swim group, which went through the swim and then the EPM. The other group is a control group that did not go through the forced swim, it only went through the EPM, so it's controlling for the swim. Now when we look here at the time spent in the open arms, again, we don't see a difference between our two groups, but when we look at the entries into the open arms between our control and swim groups, we do see a significant difference between the two groups, with the control mice entering the open arms on average about 4 times more. So this is pretty interesting. It basically showed that we were able to produce this stressful phenotype in the mice because the mice that did not go through the swim were less stressed and enter the open arms more times. So following this, we wanted to use the second of those two technological approaches that I mentioned, and this was called designer receptors exclusively activated by designer drugs, or DREDs. These are basically special engineered G-protein couple receptors. And the ones that we use were inhibitory receptors that are exclusively activated by an inert ligand. In this case, the ligand was clozapine oxide, or CNO. The idea here is that I'm able to, again, stereotaxically inject these receptors into the VNST of these mice. And when I then systemically inject CNO into the mice, when CNO binds the receptor, it causes hyperpolarization and decreased excitability of these pre-expressing neurons. And again, those are the Dynorphin-expressing neurons as well. And this basically serves to functionally inhibit those neurons. So we're functionally inhibiting the Dynorphin, again, in the VNST. Again, I performed a set of control surgeries here as well, where I just injected a control virus to control for the surgery itself. Now, this is actually some behavior that I just ran on Wednesday. And what we're looking at here is the time in the open arms for these mice that went through the same behavioral paradigm that I mentioned most recently, where we put them through the forced swim and then this time after a 30-minute break, ran them through the elevated plus phase. So now when we look at the time spent in the open arms between our control group and our DRED group, we don't see a significant difference in the time spent in the open arms. We do have a small sample size, and we're curious to see if we can see a significant effect with a larger sample size, but we can't make any conclusions off of this figure here. But again, when we look at entries into the open arms, we do see a significant difference between the control and the DRED groups. Now, this difference is actually the opposite of what we expected, because remember, with the DRED groups, we're functionally inhibiting Dynorphin neurons, and we expected that that would lead to a less stress and less anxious phenotype. But it seems that the functional inhibition of Dynorphin is actually having an anxiogenic effect. Now, it's important to note that this is dealing with Dynorphin in the VNST. And early on, I had mentioned previous studies have looked at Dynorphin and the Dynorphin core system systemically. So this seems to be contrary to a lot of what's in the field, but again, to emphasize, it is preliminary data, and this is looking specifically at the VNST. So moving forward, we definitely want to increase the sample size, basically run a lot more mice through the DRED surgeries and forced swim EPM paradigm that I've just shown you. And we also, in general, know that what we're working on here is really one piece of the larger puzzle of being able to understand some of the circuits involved in these anxiety disorders and stress response could be a great first step towards being able to better elucidate some of the mechanisms underlying the stress response and anxiety disorders. With that, I'd like to give a huge thank you to everyone in the Kash Lab for guiding and supporting me through this, and especially Nikki Kapik, a graduate student in the lab who's been a phenomenal mentor for me throughout all of this, and also to Lexi, who's been great at showing me the ropes with a lot of these methods as I've been learning. I'd also like to thank my biology research sponsors, Dr. Lohman and Dr. Burmeister, of course Dr. Bruno for leading us through our honors class here, and Summer Montgomery for doing a lot of the administrative and logistical work for us for this class and for the department. So with that, I'm happy to take any questions.
Yeah. Nice job. So if you did the channel rhodopsin injections, I'm just wondering if you have done any experiments where you drove those neurons with channel rhodopsin to see what happened. Sure.
So opto experiments, basically.
Yeah.
So we had done some opto early on, but didn't really follow through a lot of stuff. And I think it will be the next step to follow up with that. Some of the early opto work we did actually was consistent with with the DREAD data that I just showed you, but there is a lot more work to be done with it. So I mean, that is an exciting next step. Anybody else? All right. Well, thank you very much, everyone.